In this video, I will show you how to extract DNA from bacteria growing on the edge of a lettuce leaf using the Nucleospin Plant 2 kit. Here's the protocol for that isolation. We're skipping step 1, homogenation, moving on to step 2, popping the cells or lysis of the bacteria, followed by filtration, binding of the DNA to the column, washing the DNA, and then finally elution of the DNA. I've labeled a few tubes. These are microcentrifuge tubes. I've labeled them L and S for the 2 samples of lettuce that I have in this bag here. It's nice and ripe in there, so I left that out for a day just to get some good bacteria. We're going to be performing these in microcentrifuge tubes, as you saw there, that are sterile. And we're going to be using pipetters P1000, P200, there's another P1000, P10 and P20, different types, sizes of pipetters to move the volumes and reagents throughout the tubes. These are pipette tips that are also sterile with barrier tips. Over here I have a block heater that's set at 65 degrees, which is what we're going to use to pop the cells with or incubate the bacterial cells from this lettuce that I have here in this bag. So I did let this get a little ripe. You've probably seen little bacteria, black bacteria on the edges of, as you see there, of lettuce. Now this one is definitely pretty juicy at this stage, but if you've ever purchased lettuce from the supermarket, sometimes you see the edges looks a little bit black. So the question is, what is that bacteria there? Is it one species, or are there a lot of species there in that bacteria or in that lettuce edge? This is a green leafy lettuce that we have here. So I'm taking 2 samples of this to mostly just to balance the centrifuge, because this requires centrifugation through part of the extraction process. So in the second tube, I'm actually putting a little bit more of sample just to kind of test a little experiment here to see which one works better downstream, which means later on with the DNA extraction and the genome sequencing, actually, the Oxford Nanopore genome sequencing. So they're sitting in a rack. That's called a microcentrifuge or Eppendorf rack in the olden days. So that's kind of what you work in and out of. I'm changing my gloves because I don't want to have any crossover contamination from my dirty gloves to my pipetters or any of the tubes, the rack, or any of the reagents that I'm using. The first step is to add the lysis. Lysis means to pop. This is going to help to pop those cells so that we can get the DNA out of the bacterial cells. And this is the PL2 buffer. We need 300 microliters, so I'm using the P1000 pipetter. We need 300 microliters from that into each of these tubes. So you can see the tip is hovering above the first microcentrifuge tube, about 1.5 centimeters. The second tube, I just stick it down in there because this tip is going to get trashed. You see my hand disappear. I'm trashing it in a trash can down there. Then we're going to add 10 microliters of RNase A. And RNase A, the purpose of that is to digest any RNA that is in those samples. We want to isolate or extract the DNA, but we really don't need that RNA in there. So it's 10 microliters of RNase A that will go into those tubes. So I've got a P10 that I've taken out. I put on a new tip. I put that right down in the tube, and I change the tip. Now I'm getting another tip. 10 microliters, and just making sure visually that it goes down into the microcentrifuge tube. You always close your stock before you move on to closing the actual samples. So here I am closing the samples, and now I'm going to shake these samples up so that the PL2 buffer and the RNase is well mixed. So that's what it looks like. So now I'm going to walk these over and put them into, this is next to my dining room. It's COVID-19 time still, so I'm working at home in the dining room right next to the kitchen on the table. And they're going into this block heater, which is at 65 degrees Celsius. And they're going to sit there for 10 minutes or more. And so, just like a good cooking show, the 10 minutes is up in the video. And so you can put this in here for 10 minutes or an hour, 3 hours, but 10 minutes at least to help pop those cells with the heat. There's a detergent in PL2, and then you also have some RNase in there chewing up some stuff as well. Well, the RNA. So then they get placed into the rotor inside the centrifuge. Put the lid back on. You can see that those tubes were balanced. They're opposite of each other in the centrifuge. And we're running this for 2 minutes at 13,400 revolutions per minute, or RPM. The 2 minutes is counting down, and it's up. So the centrifuge is ramping down. It's going to stop. Take the lid off. There's that balance that you see, one on each opposite sides. Now the purpose of this centrifugation here, it's not actually in the protocol. It's to separate out all that cellular debris from the actual liquid. So the centrifuge spins at what we had it set at 13,400 RPM. And you can see in that last sample, it pulled down all that material to the bottom of the tube. And we have a liquid on top called the supernatant. Now I'm showing you what the columns look like that's coming up in the next step here in a moment. But the first thing we need to do before we put our sample into the column is we are going to go ahead and add PL3, 300 microliters of PL3 to these samples. But before we do that, we have to pull off the supernatant, which is the liquid on top. And it's hard to see there with my hand in the way. But we want to pull off that 300 microliters, it's about 300 microliters of supernatant on the top, and trash the cellular debris and anything else that's spun to the bottom of the tube. So that's what's happening now. So I'm pulling off the supernatant, the liquid on top, trying hard not to pipette up any of that pellet or cellular debris at the bottom of the tube. And I'm throwing away that leftover as well as the tips. So that's about 300 microliters, give or take. And now we're going to add this, it says precipitation buffer, PL3. We're going to add 75 microliters of PL3. I put it at 65. Not sure why I did that. So that's actually incorrect. You want to use 75 microliters. Hey, but that's how it goes. I didn't see that. So what's it going to precipitate? This is going to precipitate any of those carbohydrates that are in the cell wall. So there's a bunch of carbohydrates, lipopolysaccharides, slimy stuff in the cell wall of those bacteria. And we want to precipitate, which means to bring out a solution, all that slimy material part of the cell wall. So you add that PL3 buffer. I don't know what's in there, but I'm assuming it's potassium acetates. That's normally what's been used the last 50 years to help precipitate those compounds. Then you're supposed to put it on ice, and so here's my freezer full of junk and kind of dirty. You're supposed to put this on ice for 5 minutes, and I didn't really have any ice. I got some ice packs, but I went ahead and just stuck it in the freezer there. Okay, 5 minutes is up. And you're seeing it's kind of thick now, pulling that out. The cold temperature helps to precipitate those that slime in the cell wall. So there's some hydrated polysaccharides, and like I said, lipopolysaccharides in there we want to get out. The next step is to filter out any of those things that precipitated as well as anything that crossed over after we did that last pipetting. So now we're going to take out all of the liquid and put it into the violet colored column. So this is for filtering here. So there's the one, and I closed it up. There's two columns that come with the kit. There's the violet colored and the green colored column. The violet is for, as I said, step number 3 in the manual filtration or clarification of the sample. So anything that is large or precipitates is going to be trapped on the top, and whatever goes through that column, it's already dripping through and goes down to the bottom of the collection tube. That's what we want. We want the clarified material. Okay, so with one hand there, it's not easy to do. See me opening up the centrifuge again, placing the tubes opposite each other so it's balanced, closing up the microcentrifuge, and spinning it. That's full speed, 13,400 RPM for 1 minute. So there you go, there's the 1 minute is up. And you'll see there that the liquid went through. Just putting those back into the rack, making sure to keep the proper order. And next we're going on to the binding buffer, or what's called the PC buffer. And for this step, we need to discard the column, the purple or violet column that was on top. And we need to add that binding buffer, which is a concentrated salt. And we're going to add 450 microliters of PC. So it's a guanidine salt in very high concentration to each of these tubes. And then they're going to be mixed and added to the new column that's right in front of them, which is a green column. You can see me hovering again above the collection tube, not to touch it so that I contaminate the PC buffer. And then I lose the tip. So DNA will bind to silica, which is glass. DNA will bind or attach to silica in the presence of these guanidine salts, a very high concentration of the salt solution. So that was added to the sample. It's being mixed, and that's going to be pipetted into the new column, which is a green column sitting inside of a collection tube. And the idea here is now, rather than that collecting what flows through, we're actually going to keep the column there, and we're going to collect what binds to the column. So I just pipetted in that sample. Now I'm going to mix and pipette in this sample. And so this is the binding step, step 5. We want to bind the DNA to that column, which is made of silica, during this step. Now I wasn't paying too much attention to my volume, so the first collection, or the first tube, I actually put a little bit too much volume into. And so it says don't put more than 700 microliters, but I wasn't paying attention. And so this one I fixed it, made sure it was 700. If you put more than 700, what happens is when you close the lid, some of that liquid might kind of squeeze out, which is not what you want. So I kind of noticed it right there. So that's what I'm doing. I'm going to the kitchen to get a paper towel to get a little bit of that liquid that overflowed there. It's a rookie mistake. I've probably done, I've been doing these extractions since the 90s. I don't know how many thousands I've done, but this is a new kit for me. So I'll just blame it on that. It's a little new. So then you take those samples, their green column, put them in the centrifuge opposite of each other, and we're going to spin for 1 minute at, in the manual says greater than 11,000 G. I'm running it at full speed, 13,400 RPM. To calculate G based on that RPM, you need to know the size of the rotor and distance from the sample to the center, and I never measured that, so I'm just going to ran at full speed. So these are all done. You can see the liquid, what's called the filtrate, went through, and now the DNA is bound to that little white portion where the green is. Okay, so the liquid was going to be poured off if I didn't do it already. And now we're going to wash the DNA, which is bound. Oh, I didn't pour it off yet. Now we're going to wash the DNA, which is bound to that silica membrane. So I discarded, you keep the same collection tube, discarded the flow through. And this is where something else pops up that I see. You see how there's some liquid still stuck in the column? That's a problem. So all the liquid had not gone through, probably because there's quite a bit of polysaccharides in there that are gumming up the column. So it always helps to look at your samples. And so I'm actually putting these back in to spin again real quick, because there's still about 40 microliters, it looks, in that second sample that I labeled S. And so I'm going to spin this for a tiny bit. Looks like 33 seconds out of a minute, 27 left, just to get that tiny bit of liquid through the column. If I didn't put that liquid all the way through the column, then when I add the next step, it might actually cause some problems to the following steps or downstream process. So it's good to look at your samples, know what you're doing. All the liquid should be through, and that white membrane, that silica membrane, you should be able to see it, and it shouldn't be wet, it should look basically dry. So now you can see that it's actually gone through sample number 2. How often does that happen? Occasionally it happens if you've got some really gooey samples. So why did it happen to this one? Because I put extra sample into that tube. That's why. So there's extra inhibitors in there. So now we're going to add 400 microliters of wash number 1. And so here we go. Wash number 1. This is an ethanol solution. And the purpose of really both of these washes, I'm going to do 2 washes. The purpose of both of these washes is this wash step is to remove all that salt that we shoved in there. There's tons of salt in there, and I want to get that salt out because that'll affect what we do with this DNA when we're done with it. So the tubes go into the centrifuge. First wash, 400 microliters, called PW1, 1 minute, full speed. So you can see that the liquid, the 400 microliters, went through there, and it actually looks like it did a little bit of cleaning for us, or wash for us. You can actually see the liquid now has a little bit of pigment in it. So it's doing its job. The purpose here is to wash that DNA as it's bound to the silica. Now we're going to go to the second step, which is the PW2 wash buffer. You have to add the ethanol to it when the kit arrives, so you see I've marked that. What we're going to do is we're going to discard the flow through from each of the tubes. That's the 400 microliters from the first wash. You can see the nice green column there. So the DNA is bound to that silica in the column. And now we're going to add 700 microliters of this PW2 ethanol wash again. Slightly different wash buffer. And there I go, balancing act. Why doesn't the DNA come off the column? DNA is not soluble in ethanol. So we're washing this in a high percentage of ethanol solution. It's not soluble in ethanol, so it stays bound to the silica membrane, even though we're adding liquid onto it. Okay, so I had a little bit of overflow again that I didn't like to see on that second sample there. So I went and got a paper towel and just washed that because I didn't want it going into my centrifuge. Each time I perform a DNA extraction, I clean that rotor. That's the black disc-like thing that you see there. But all that salt gets, you can see a little bit on the edges just from what we did here. All that salt kind of spins off the lid a little and gets in there and can be contamination. Okay, so this is PW2, 700 microliters for 1 minute, full speed. So it's looking clear. The liquid has gone through. That was the second wash. There we go. You can see there's a lot of liquid in there. So what the protocol has you do is it has you use the PW2 wash one more time, and you're going to discard, just like before, the flow through because the DNA is still bound to the column. But this time, rather than adding in 700 microliters, the protocol calls for 200 microliters of an additional wash. So there's the 200 microliters. We don't want to put another 700 in there because you'll see that the bottom of that column actually has some ethanol, residual ethanol on it because it actually touches the column. So it's just 200 microliters to spin through there one more time. And that 200 microliters is so far below the column that, just a second, it'll come back. It's so far below the column that it will not touch it. And so that ethanol we don't want going to the next step because it would inhibit that next step, which is the elution step. My cell phone fell twice. Okay, so back over to the centrifuge. 200 microliters of the second wash buffer, PW2, and this is going to spin full speed for 2 minutes to make sure, I have it set for 3, to make sure that that ethanol goes all the way through there and there's no residual ethanol left over. I don't think I ran it all 3 minutes. I think just for fun I ran a little extra, so you can see there 35 seconds left, so 2 minutes and 25 seconds. The other kit I use actually does 3 minutes, so that one I've used since 95 or 6, so I tend to want to run it a little bit extra on this one. So you can see I'm being very gentle, taking these out, so that ethanol on the bottom does not splash up and touch the bottom of the green column. Right in front of me I have 2 microcentrifuge tubes labeled the same, L and S. These things come sterile, DNA free, so they're all ready to use. You're going to discard all that flow through and the collection tube and place the column into the microcentrifuge tube. Because now what we're going to do in the next step here is we're going to add what's called an elution buffer. There it is. So it's pH calibrated, it's got a little salt in there, but it's basically just water. This is sterile water, and we're doing 50 microliter elution. So you take 50 microliters of this water buffer and try and pipette it directly onto the silica column in the center of the white column. This is going to set for 7 minutes. So DNA is soluble in water. So DNA is going to come now into solution. And after 7 minutes of sitting there, we're then going to centrifuge these samples. So I'm closing up my source here. Put that out of the way. The 7 minutes are up. Grabbing the 2 tubes. And now I'm going to centrifuge them because we need to pull that water and the DNA through that membrane and down to the tube. And that will be our purified DNA. The kit calls for incubation of that DNA for 7 minutes. It calls for incubation at I believe 65 degrees Celsius. I don't incubate my samples like that. I just do the room temperature. So that was the 7-minute incubation followed by centrifugation, and there is the 50 microliters in the bottom of the microcentrifuge tube. And so that is ready for the next analysis, which is the genome library. That's ready for the processing, or you could use that for a whole bunch of different molecular biological techniques at that stage. So you take the column out, you can throw that away, close the lid, and you're done and ready for the next step.